Welcome back to the lab. In our last episode, we learned how to clone a gene. But now that we have our colonies, how do we know which ones actually contain our new gene? Today, we're taking a shortcut to success with a technique called Polony or Colony PCR. And we're back. After a successful cloning experiment, you're left with a plate full of bacterial colonies. The problem is, only a small percentage of these colonies actually contain the plasmid with your gene of interest. Finding the right colony can be a tedious process involving mini preps and restriction digests. This is where Colony PCR comes in. It's a rapid and cost-effective technique that lets us screen our colonies for the correct DNA sequence without the need for time-consuming DNA extraction. You simply pick a tiny bit of a colony, use that as your template, and run a quick PCR reaction. We use this technique after major genetic manipulations like cloning, transformation, or mutagenesis to quickly verify our results. It's a fast-track way to confirm if your experiment was a success, saving you a ton of time and resources. Now let's get to the fun part. Here's a simple step-by-step protocol for performing Colony PCR right on your lab bench. 1. Sample preparation. First, you'll need to transfer a tiny amount of a single bacterial colony into a sterile tube containing a small amount of sterile water. It's very important not to take too much colony material, as that can lead to PCR inhibitors and a failed reaction. 2. Cell lysis. Next, you need to break open the cells to release the DNA. We do this by heating the mixture at 98 degrees Celsius for about 7 minutes. This simple heat step is usually enough to lyse the bacterial cells and make their DNA available as a template. 3. PCR reaction setup. Now we set up our PCR reaction using the cell lysate as our DNA template. We'll combine our PCR master mix, your specific forward and reverse primers, and the cell lysate. If you need a refresher on the magic of this reaction, be sure to check out our previous episode all about PCR. 4. PCR cycling conditions. You'll run a standard PCR program, but with a slightly longer initial denaturation step to ensure all cells are completely lysed. Initial denaturation, 94 degrees for 3 minutes, 30 cycles, denaturation at 94 degrees Celsius, annealing at your primer-specific temperature, and extension at 72 degrees Celsius. Final extension, 72 degrees Celsius for 7 minutes. 5. Gel electrophoresis. Finally, load your PCR product onto an agarose gel. The presence of a band at the correct size confirms that your colony contains the desired gene. If you need a reminder on how to read these results, we've got a whole episode dedicated to gel electrophoresis. Like any lab technique, Colony PCR can be tricky, but don't worry, we've got you covered with some quick troubleshooting tips. Problem 1. No PCR product on your gel. Possible causes. This could be due to poor cell lysis or incorrect primer design. Solution. Make sure you've heated your sample for the full 7 minutes at 98 degrees Celsius. Also, double-check your primer sequences and annealing temperatures. Problem 2. Weak or non-specific bands. Possible causes. Your annealing temperature might be suboptimal, or you might have used too much colony material. Solution. Try optimizing your annealing temperature. If that doesn't work, reduce the amount of colony you use in your sample prep. Problem 3. Smearing on the gel. Possible cause. You've likely overloaded your gel with too much PCR product. Solution. Simply load less of your product onto the gel. By following these simple steps, you can troubleshoot most of the common issues and get clear, reliable results every time. So there you have it. Colony PCR is the perfect final step after a successful cloning experiment. It's fast, efficient, and essential for any molecular biologist. If you're ready to get to the next level in your biotech journey, be sure to hit that subscribe button and ring the notification bell. Our How to Biotech Lab playlist is full of more amazing techniques like this one. Join us next time as we dive into a brand new topic. Until then, keep experimenting.